Good morning, everybody. Today, I'm going to talk about my senior project progress. In the title is Analysis of Genetic Variation of the Chong from Eastern Thailand using MySeq FGX Genetic Analyzer. Let's make sure that we all remember the research objectives. The 30 individuals from Chong people from Chanthaburi province in Thailand are analyzed from this study. To study and report genetic variations of Chong people from the eastern part of Thailand with 27 autosomal microsatellites and 170 autosomal single nucleotide polymorphism or SNP genetic markers by using MySeq FGX Genetic Analyzer. From here is the research plan that all steps start on December 2021. It will be finished in April 2022. From this table, the red line is the research plan and the patch line is the finished work. For 2 months, I have finished. The first step is about the study, the relevant research and applications of the MySeq FGX Genetics Analyzer. And now I'm going to analyze the Chong population samples by using this machine. It will be finished in 3 months on February. Now we are moving to the process steps. I separated the studied research into groups. The first is study the relevant research that composed of the originated and genetic structure of Chong people in Thailand, the genetic markers for studying population genetics and applying them to use in Thailand. Last group is studied the application of the MySeq FGX Genetic Analyzer. It's about the applying the MySeq FGX and Genetic Analyzer for forensic science from the other countries. Here, analyze the Chong samples by using MySeq FGX Genetic Analyzer. From this study, 30 individuals of Chong people will be analyzed and 20 of them have finished to analyze. And here, there are 5 unprocessed steps, including the first is analyze the residual Chong samples by using this machine because 10 samples are still not analyzed. Collect the data from MySeq FGX Genetic Analyzer. Summary and discussion. Compile information and prepare a research project booklet. And the last one is present a research project. Here is the research assessment. The completed works account for 60% of the total work, and the research operation will be success according to the purposes and in its time set. And the last topic is the problem that happening in my study. The result of the genetic variations of Chong people is relatively rare. Some genetic markers were missing after being analyzed by using MySeq FGX Genetic Analyzer. This may be due to an experiment that went wrong with the protocol. It should be studied the procedure of experiment and do it carefully, follow the protocol step by step. And due to the epidemic situation of COVID-19, causing the action plan deviated from the target set and causing research to be slower than expected. I would like to appreciate Professor Ripu Gutanan for supporting this study, and thank Vipada Waravatin and Nongrak Prakhan for technical assistance and for donating this sample for analysis. Thank you for your attention. Does anyone have any questions?